Dear friends, Assalamu alaikum. In this video lecture, I am going to talk about the mechanism of contraction of the cardiac cells. It's a very interesting topic, so let us begin. Cardiac cells as well as the skeletal cells both are contractile in nature. That means they show contraction on stimulation. But there are certain kinds of differences between the cardiac cells and the skeletal muscle cells. These differences I will discuss them. See, this skeletal muscle cell gets stimulated by the neuroendings to cause the action potential, or we can say it causes the action. On the other hand, cardiac muscle cells get stimulated of its own. That's called the self-excitation. That means they are self-excitable, and this very property is called the automaticity or autorhythmicity. That means there are certain kinds of cells which are found in the heart which shows automaticity or autorhythmicity. That means they get excited of its own to show the action potential, or we can say it shows the depolarization process. On the other hand, there is one more difference between the skeletal muscle cell and cardiac muscle cell. That is, skeletal muscle cells of a given unit, of a given motor unit, contracts at the same time, and but the impulses doesn't get flow from cell to cell because it lacks the gap junctions. On the other hand, cardiac muscle cells contract as a unit or doesn't contract at all, as the flow of ions passes from cell to cell. That means the cardiac cells are adjacent with each other. They are held together with the help of desmosomes, and the flow of ions takes place with the help of gap junctions. That's the reason it gets contract when it gets stimulated as a unit, or it doesn't contract at all. That's why the cardiac cells shows a functional syncytium. And third point, that is the difference, this is called the absolute refractory period. Absolute refractory period, it is the inexcitable period in which sodium channels are closed or open or inactivated. And this absolute refractory period, that's represented as ARP, is as long as the contraction of the cardiac cell. That means that is 250 milliseconds. The prolonged ARP prevents the cardiac cells from tetanic contraction. There is a prolonged ARP, absolute refractory period in case of cardiac cells. It prevents the cardiac cells from getting tetanic contraction, as the ventricles are responsible to pump the blood out of the heart to flow that very blood to all cells of the body. On the other hand, ARP for the skeletal cell, that is 1 to 2 milliseconds, and the duration of contraction is 10 to 100. That is a peculiar difference between the cardiac cell and the skeletal muscle cell. I had discussed the dissimilarities or the differences between cardiac cell and the skeletal cell, but there are similarities as well. As you already know, I have told you that automaticity, only it is the 1% of the cell of the heart which shows automaticity. They are all called the nodal cells, which I will discuss in the cardiac physiology, or we can say when we talk about the physiological events of the heart. Here only 1% of the cardiac cells show automaticity, and the rest of the cells are contractile muscle fibers, which shows the electrical event similar to that of the skeletal muscle cells. These events are: the first point, here is the influx of sodium from the ECF. Influx of sodium from the ECF takes place through the voltage regulated fast sodium channels. The voltage regulated fast sodium channels gets opened through the opened voltage regulated fast sodium channels. There is an influx of sodium from ECF. It changes the permeability of the membrane. That means the action potential causes the reversal of membrane potential from minus 90 millivolt to 2 plus 30 millivolt, which I had depicted in the chart here. Here is minus 90 millivolt when action potential takes place, when there is an influx of sodium takes place, it gets changed minus 90 millivolt to plus 30 millivolt. What happens then? There is the transmission of depolarization wave down to the T tubule. Transmission of depolarization wave down to the T tubule. T tubule is nothing, it is actually the invagination of the cardiac cell, this sarcolemma of the cardiac cell. What happens? T tubules, we can say the transmission of depolarization wave down to the T tubule causes to release calcium from the sarcoplasmic reticulum into the sarcoplasm, from sarcoplasmic reticulum into the sarcoplasm. And then the third point, that is similar in case of cardiac cells as well as the what it called as the skeletal cell, that is called the excitation contraction. Coupling occurs as calcium provides signal. When the calcium that gets released from the sarcoplasmic reticulum, this released calcium, what it does is that it enhances excitation and contraction by providing signal via troponin. The troponin is a protein that gets binds with the myofibrils, that's actin and myosin, which helps to cross bridge actin and myosin. And what happens then? The sliding mechanism of action takes place. Then what happens? These three are the similar kinds of the actions which are shown by the cardiac cells as well as the skeletal cells. But here you must remember that how this calcium gets released, it depends upon the type of the cell. That means a skeletal cell shows different kind of mechanism to release the calcium. On the other hand, the what it called is the this cardiac cell shows the different kind of mechanism. We talk about the, let's look on this. It is said that 10 to 20% of the calcium enters into the cytosol of cardiac cell from ECF via calcium channels. These channels are called the slow calcium channels. I'll highlight this with the help of marker. Calcium channels, these are called the slow calcium channels, as they get open a bit late. How these slow calcium channels gets open? Because you know that the calcium channels are very closed. We can say the non-stimulated cardiac cells have the calcium channels. The entry of the calcium is beard, or we can say that it doesn't get enter, the calcium doesn't get enter from extracellular fluid into the cytosol of cardiac cells in the non-stimulated cells. But when the calcium, when the sodium dependent depolarization occurs, it changes the voltage of the membrane that is responsible to open the calcium channels. That is why they get we can say open a bit late. What happens? It stimulates in turn the SR. SR means sarcoplasmic reticulum to release 80% of the calcium. That means 10 to, we can say 20% of the calcium comes from outside into the interior of the cardiac cell. Then the 80% of the calcium is released from the SR when it gets stimulated by the calcium which comes out, which comes from the outside of the cell. I hope that you got this point. 80% of the calcium gets released from the sarcoplasmic reticulum via special sensitive channels called the ryanodine channels. Must remember, there are ryanodine channels which are found on the membrane of the sarcoplasmic reticulum. Through this, the calcium gets out from the sarcoplasmic reticulum in the form of sparks, that's called the calcium sparks. And what happens? There is a very high concentration, we can say the rapid concentration, or we can say dramatically increased cytosolic calcium level inside the cardiac cell. Then what happens? At this point, sodium permeability reaches to its resting potential. That means depolarization begins. As I have already told you, sodium channels, when they gets open, they we can say they quickly gets closed as well. The At this point, sodium permeability reaches to its resting potential and repolarization begins. But increased surge of calcium, which are found in the cytosol, causes the or it causes plateau. It causes plateau, PLATEAU. This is the plateau. I'll discuss this figure after some time. When there is an increased surge of calcium, that causes a plateau as it prolongs depolarization potential briefly. The raised level of the cytosolic calcium causes plateau, or we say it prolongs the depolarization potential briefly. At the same time, decreased potassium this permeability at the same time decreased potassium permeability also increases or prolongs the plateau and prevents rapid repolarization. At about 200 milliseconds, what happens? Slope of the AP, that is the action potential here, slope of the AP, I'll discuss this later on. Slope of AP drops rapidly due to closure of calcium channels. Calcium transported to the ECF, then once again I will tell you, slope of the AP action potential drops rapidly due to the closure of calcium channels. The calcium gets transported to the ECF, extracellular fluid or extracellular space, or into the mitochondrion, or into the what it called as the sarcoplasmic reticulum. Meanwhile, opened potassium channels, on the other hand, there are the calcium channels which are open, which are responsible to flow the potassium ions from cytosol into the ECF, that is responsible to restore the resting membrane potential once again. Another point, during repolarization, calcium is pumped back into the ECF and sarcoplasmic reticulum, and as long as the calcium enters, the cells continue to contract. Let's now look on this figure. This is the time taken, this is the membrane potential. This is minus 90, this is plus 30. What happens when there is an influx of sodium? What happens? There is a reversal of membrane potential, that is from minus 90 millivolt to plus 30 millivolt. But there is a rapid closure of the sodium channels. What happens? There is a slope, there is a brief slope, but there is an opening of calcium channels and they remain as such. That creates what it called as the we can say in the figure that very line indicates a plateau. It causes a plateau. Plateau is actually, it is the this tension in the muscles, tension development takes place in the plateau. This is the this figure indicates the ventricular cell, and there is a formation of a we can say the horizontal line, that is called the plateau. That very plateau is what it called is the gets developed due to the opening of the calcium channels. And on the other hand, the what it called as the decreased permeability of the potassium also enhances the plateau formation. But when it reaches to the 200 milliseconds, the slope drops. We can say it drops drastically or dramatically, or we can say it what it called as it drops. Then what happens? It downs. Then what happens here? The calcium goes what it called the replenishes back into the mitochondrion or the sarcoplasmic reticulum or into the ECF from the cytosol. And this diagram shows the dark line as well as the red line. This shows the relationship between the ventricular cells and the arterial cells. That means the action potential here, then the plateau formation, duration of contraction. But there is a difference in the plateau. The plateau formation is found in the ventricular cells, as they have to pump the blood out of the what it called the ventricles to all cells of the body, while as the arterial cells show the short this plateau formation. I hope that you have got the information regarding the topic, and I hope that you will you have enjoyed this video. Please share this video with your friends, and so that they can also get information regarding the mechanism of contraction of the cardiac cells. Thanks a lot.